chemical engineering, so probably completely different. And I had a chemical engineering, a minor in biochemistry, and I was working computational biology a lot, and I got interested in physics because that seemed to explain everything that's there. And so I started a graduate program here at UFM in biophysics. I work with Chris Miners, he's in physics, and so we basically try to look at how DNA plays an important role in biological functions. Okay, that's like the pressy version of what it is.
We read the review of one of your papers a few weeks ago, so we have a small idea.
So I'm Robert Deegan. I'm an assistant professor in physics and complex systems. And I started off in physics, did an undergrad at University of Massachusetts at Amherst, did a PhD at University of Chicago, did a postdoc at University of Texas, then I had a faculty job in the mathematics department at Bristol University in the UK, and then I came here. And I'm an experimentalist, meaning I work in a lab, and primarily I'm interested in dynamic phenomena, particularly where there is some flow of mass or a temperature gradient. I call it, it goes by the general name of pattern formation, and that's a good name. Basically, when you're out of equilibrium, structure develops, and I, in general, want to know how that happens.
I think we've heard a little bit about optical tweezers and quantum dots from the Miners Lab. Can you explain?
Okay, so there are two things in biophysics. One is you can go for the technique-based, and other can be the physical processes that involve. So what we try to do is we try to actually look at we are trying to actually be both. You can look at a completely biological process using a physical technique, or you can look at a physical phenomena using different biological techniques. What we try to do in our lab is trying to see how mechanical properties of DNA can actually be used to control biological functions. Everyone here knows basic biochemistry, right? Okay, so the idea is if you want to look at any proteins are thought to be the workhorses of the cell, yeah, and so a lot of protein functionality comes from how genes start getting activated. So there are proteins like transcription factors that bind to genes, yeah, and you can have activation or repression of genes. But let's say one way in which these factors can actually play a role is like a protein can actually bind distal ends of a DNA, bring them closer. Yeah. The other way to think about it as a physicist would be like so how is that you have a long piece of polymer? How is that it can get twisted and form like a loop, such a way that the protein can form like a flow. So you spend energy and so on. So we try to actually use different physical techniques to actually look at these looping or these events. One technique is actually optical tweezers, in which we try to trap a bead and see if there is an effect of force on all these biological processes like looping. The other thing is fluorescence microscopy. So basically, we try to tag along. We try to look at correlation between end motions to see if it is in a loop state or in a non-loop state. So these are a couple of my projects. So that's why I can talk about this. I can talk about it in particular, but I don't find that as a particularly of interest to general physicists. So, but yeah, if you're interested in more detail, I can actually talk about it.
Thank you. And Dr. Deegan, I think last time you were here, you explained that one of your projects was why a ring that has a bubble that has a solute in it, when it dries, leaves a ring.
Yeah. So you would like me to explain again?
Yes, please.
Again. Okay.
New people.
Out of curiosity, what the class is physicist, biochemist. What kind of class is this?
This is physics of the life sciences. So
physics of the life sciences. Okay. So so one of the things I did actually is a long time ago now. I looked at what became known as the coffee stain problem. And if you have a surface like this one, you put a drop of stuff on it, of water, and it has some particles in it, what you find is that when all the liquid is gone, all the particles end up exactly on the edge of your drop. And the question was why? And it turns out to have a really simple explanation. The liquid that's on here, and we're actually talking small drops, but I'll pretend it's big. The liquid, as it evaporates, you might think it shrinks, but it doesn't. It actually stuck at the end. It's stuck on its perimeter. Instead, what it does is it flattens down. And because the water is stuck here, as it evaporates, water leaves from here, but it's still wet here, so water must come in. So because the water is coming in, it takes with it whatever's in there. And whether that's coffee, little bits of plastic particles, which is what I did a lot of experiments on, or anything else. You'll see these in the winter. When you walk around after there's been snow, you'll see these circles of salt. And that's exactly the phenomenon that's going on. Yeah.
How did you monitor that?
Fluorescent microscopy, actually. You can buy little plastic particles from there's a whole slew of vendors, and they'll put whatever dye you want in them, and it's a very easy way to monitor them. And so actually, I spent a lot of time simply counting how many would arrive at the edge with a microscope. So that was one way we monitored.
When you were moving around your different positions, was it basically the same research that you continued to do, or did you move around from topic to topic and then finally arrive on, well, that's not what I wanted to do, I'd rather move on and then end up where you are now?
No, so in my field, one never works on the same thing when you move. And at some level, that's the seamy side of science that is driven by money. Somebody gets money to solve a particular problem, or they claim that they will solve it. And then that gives them money to hire graduate students, postdocs, or whomever. And so each time I move, I do that because somebody has a position like that. It changes when you're a faculty, then you get to choose what you do. But
Were they far apart from what you really wanted to do, or were they all a little?
No. Otherwise I would do it. These are not the greatest paid jobs in the world, and hauling. I have a family, and hauling my family across the world is not ideal for them. So there has to be something in it, and the something is usually that the job that I would do is very interesting to me.
I guess I think you can speak better for it, but then you build on your strengths. You know what you know what your strengths are, and you just sort of develop on it. For instance, for you, everything will have an underlying theme. That's what I figure. I've had 4 or 5 projects till now from undergrad, but everything has the underlying theme of dynamics of molecules. So I think there's that recurrent pattern that is there in all my research. It's sort of, I think at some point you just figure out that's your interest. Something you just like doing something. Maybe it's still because I'm naive, because I haven't gotten any of my own funding, but I just want to get somewhere else. But yeah.
So I agree with that.
Questions? Or maybe can each of you tell us what portion of your day do you spend doing any particular task? Comparing between grad student and faculty.
Okay, it'll be completely different.
Yeah.
I don't have a structure to my day. It's so there's a vicious cycle. So one day you work probably till 3 a.m. or something, and so you get up late, and so you work. Okay, since you're awake, you can't go to sleep, and so it goes till 4, and then you go to. It's a cycle, and you just go to crash. But that's not always, because at present I'm writing my thesis and so on, so I don't really have any classes or anything. So I generally meet my advisor. Sort of my advisor is flexible on when he wants to meet me. I can just drop into his office. So So in that way, I don't have much of a structure, but ideally, I generally come to lab like around 10 to 11. That's the ideal situation. 10 to 11, and I leave by like 8 o'clock. Okay, there's always regular coffee breaks and regular time for other stuff and everything, but yeah.